Hello, welcome to some video notes. Today we'll be going over section 9.3, which gets into how growth is regulated and controlled in plants. As you've probably noticed, something along the idea of tropism or phototropism before in plants, the idea that plants tend to grow towards sources of light. Also the idea of like gravitropism, or the idea that they seem to detect gravity and they know to grow down. The roots will go down where their stems and leaves will go up towards the light. So we're going to talk about how that type of growth is controlled in plants through a very special family of hormones called auxins. So it's going to be a relatively short one, just getting into how growth is regulated.
You have failed me for the last time.
So growth in plants comes from what we call the meristem. And so the meristem is either the very tip of a branch or a stem of a plant, or also there could be a section like this at the very ends of the roots, and they're what we call the root tip. And the meristem is where we're going to find essentially some undifferentiated cells, like stem cells of plants, that can kind of diversify into other different types of plant cells as that part of the plant continues to grow. And then we're also going to see some specific types of cells that are there just for regulating the development of that specific tissue as it either changes into a branch or maybe changes into a flower or a leaf, depending on what type of stem part that we're looking at, or helps protect the roots as the root continues to grow through the soil. For example, maybe in the past you've looked at something called a root cap. All right, at the very end of the roots, where there's lots and lots of mitotic division, lots of cell growth happening, there's kind of a protective layer on the very end of the root that's dead, dried out cells that it's really there to like be the battering ram that forces the root through the soil as the roots continuously grow. So anyway, regardless of we're talking about the roots of the stems, meristem is where we're going to be seeing this growth occur. And there are different types of growth depending on where or what type of direction we're going. So we have apical growth, and apical growth basically is up and down. That is like a vertical growth. So that's the roots going down into the soil or sideways into the soil as well, or branches and stems coming up out of the soil or branching sideways above the soil as well. And we also have lateral growth. And so lateral growth is when the stem and the branches and the roots get thicker in order to deal with the increased mass of the plants, right? So you can imagine like a giant tree, right? So that tree, really great source of lumber for us for building and things like that. It has this massive trunk, that in the middle that we could be cutting down and using as a source of wood. So how did that get so big? Remember, all living things would have originated as a single cell. So it would have started as a single cell, and then it would developed into, the seed. The seed would eventually developed all the roots and the initial stems and leaves. But eventually, as that tree gets longer, if it's going to support its weight, it's going to need to get thicker in order to be more stable. So the roots are going to have to increase in size as they spread out. The stems are going to increase in size as the tree and the bush continues to get larger. So that type of lateral increase or thickness is coming as where layers are added on in the Cambium layer, and we'll look at transverse slides again to show you what this looks like. And so if you're thinking about trees, by the way, you're probably maybe thinking of like rings on a tree, like how we can count the ages and how many years the tree's been around before it was cut down. That's what the kind of lateral growth we're talking about. So every time one of those new rings is added, it's added through lateral growth. That's how big the stem got or the trunk got between each year as the distance between those rings. Okay, so regardless of what's happening, growth in a plant is going to happen in different ways. First off, we could enlarge the cells, which means that we elongate them. We make longer cells than what they originally were. And then we could also just increase the number of cells, right? We could do lots and lots of mitosis, and so that does a lot of cellular division. And so eventually more cells in an area is going to cause that to kind of stretch and elongate. It's either going to cause it to get thicker using lateral growth or get longer using apical growth. right? So these are the two different types of growth that will occur. Often we will see a cell enlargement, and then eventually once the cell gets so large, we will then do mitosis, right? Because remember, we have to maintain a good surface area to volume ratio to have nice stable cells and have them maintain homeostasis. So regardless of the type of growth, this is where that's how the growth occurs, either enlongment of the cells or through rapid mitosis. So before we get into how this growth is regulated, first let's just go through the specifics of what we mean when we talk about apical growth versus the lateral growth. And so apical growth occurs at the meristems, right, at the very tips of the stems, or the very tips of the roots, right? And so this is where what we call a terminal bud, right? And so this is where or the root tip, if it's in the roots, it's called a root tip, or the stem terminal bud. And this is where we have all of those meristem cells, right? We have the stem cells, basically, that can diversify into, well, maybe this is going to branch off and bud off and become a leaf, or maybe this is going to develop into a flower, or different types of materials, different plant tissues can come from this. And so it's from this apical growth that we have this increasing in length and the size of the plant, right? And so in the section of this growth, we get this internode in these auxiliary buds, so the very, very ends of the stem where we can start doing growth sideways, right? We have a bunch of, the plants have a bunch of inactive meristem tissue. And so really what the plants wants to do is that they want to try to grow up as much as they can, right? Because really the sun, they've figured this out, is above them, right? And so the taller they can get to a certain point, their maximum height, the better chance that they're going to be able to, spread themselves out and be able to, get as much sunlight as possible for photosynthesis. So plants will typically grow up as much as they can, as much as possible, before they really start to grow outwards. They can kind of do both at the same time, but their main focus is to grow upwards. And so their horizontal growth is going to kind of be inhibited for a little bit while the vertical growth is taking over. Then once that vertical growth is either at its maximum, so that the maximum point that plant itself, based on its species, does grow, or when it hits some type of barrier, then it will actually start to grow out sideways. And so we'll get horizontal growth out of the little buds along the stem. And so this area here, to kind of help circle this, so this area here will keep growing north, or keep growing up, not north, keep growing up. And eventually, when it either hits its maximum or it's inhibited and it can't grow up anymore, that's when things like this are going to start kicking into their growth. And so their little sub-branches, little nodes for horizontal spreading, are going to start becoming more expressed in order to start growing in other directions. And so this is something that farmers can actually utilize a lot if they want to kind of control the size of their crop plants, depending on what their overall goal is for how they've been placed in a garden, or if they've been placed in a specific farmland. So what happens if the plant gets to a certain height and they want to stop it from growing any higher than that, so this is something like gardeners will do a lot, they will come along and they just cut off this apical stem, right? The meristem at the very top of the terminal bud. And by doing that, by cutting it off, the plant thinks, we've hit our maximum. We can't grow up anymore. So then it will start to grow out sideways and do more horizontal growth because it's no longer doing its lateral growth. So we can actually influence the growth regulation of these plants by artificially either inhibiting the movement of the meristem at the terminal bud, or just removing it entirely, just cutting it off, can then cause the inhibition of the horizontal growth to be removed, and so the plant starts spreading out sideways in order to spread its leaves as much as possible. Okay, so that's the horizontal growth, by the way. So now we're going to look at the lateral growth. So that's the the apical growth, growing up and elongating sideways is what we mean by apical. So now we're going to look at lateral growth. So lateral growth is this idea of increasing in thickness. And so here we've got a transverse section of a slide of a stem, right? And so there are some things in here that you should already be able to identify, right? So we have they're calling it this cork outside section, but really that you could also think of that as the epidermis, right? That dried outer section of the plant, right? Then we have our cortex, where they're calling it the cork cambium here, but that's the cortex as well as what we've labeled it earlier. That's that tissue layer between the epidermis and the inner layers of the plant. And then, of course, we have the phloem, which is going to be on the closest thing to the epidermis, which is part of our vascular bundle. There's our cambium strip, which is that layer between our phloem and our xylem. And then we have our pith down here, right? And so this is not as typical as the ones we were looking at before. We were looking at kind of like oversimplified versions of stems earlier when we looked at phloem and xylem and vascular tissues, because it's very clear to see like just the different types of xylem versus different type of phloem. But now we're looking at a type of plant which goes through seasonal lateral growth. And so it looks slightly different than what we looked at before, because we're actually seeing the results of different seasons of growth. And when that growth occurs, we're going to see an increase in the amount of phloem and an increase in the number of xylem, all right, in order to increase the movement and the transportation through transpiration and translocation. If we have a bigger plant, like a tree, like if the tree is going to get bigger, it's going to have more cells. So it needs to have increasing the ability to transport water and the ability to transport sucrose solution around the plant at a faster rate. So in between the cambium and the pith here, all right, you see some areas, right? So we've got extra layers of phloem, and we've got, if this was the original xylem here, right, that's our original xylem, we've got extra xylem that have formed, actually, sorry, reverse that idea. If we have our original xylem is here, and then we've got this new xylem that's formed closer to the cambium. So from the cambium layer is where these new tissue layers are going to originate, right? So this cambium strip, this divider between the phloem and the xylem, in addition to being a connecting point between these two types of vesicles so that they can help with the movement of water and the movement of sugars throughout the plant, it's also where this new growth is going to occur. So from the cambium towards the epidermis is where we get a seasonal growth of new phloem developing as we do lateral growth. And then the cambium towards the pith, right, is where we get new development of our xylem. All right, so our extra xylem, our new developed xylem, in order to, again, increase the movement of water efficiency as the plant increases in diameter, as it does lateral growth. All right, so plants grow taller, but they also need to increase in their mass. They need to increase laterally. So they need to increase the amount of xylem, or what they call secondary xylem, and they need to increase the number of phloem, or their secondary phloem. So they have our secondary phloem, and here is our secondary xylem, which have appeared from that cambium layer, right? And so every time this happens, a new cambium layer is kind of created as we go through this rapid growth. And so then we start creating these rings of new xylem and new phloem around this cambium layer. And then it goes completely around, the stem or the trunk of the tree. And so every time the tree goes through this growth, every season it goes through rapid growth. Normally this is the spring and summertime is when it commits a lot of energy to this rapid growth. Well, rapid for plants. It's not, rapid like an animal does, but the growth, of the diameter of the plant, we get these new layers, and those new layers make that new ring. And so that's how we can count the age of a plant, or the age of a tree, for example, by counting these cambium strips that have been created and the xylem and phloem layers around them. Now, it doesn't keep all of them, right? Eventually, it gets to a point where it starts to increase in size, and because of all this extra weight, it starts to be, it's not going to be stable having all this functioning tissue inside of it, all this extra water and sucrose moving through this tissue. And so as it increases in size, as it has more phloem and more xylem, eventually some of the inner tissues are going to become more like pith, and they're going to dry out and just become wood material. And so they become dry, dense material on the inside of the tree, right? That's the wood material that we like to use for our building. And then it's purely there for structural support. right? And so it does, as the plant's going through rapid increase in development, it needs more phloem and xylem to help with the efficient transport of sugar and water. But then eventually there's this nexus point where eventually it gets so big that some of that extra material, some of that xylem in the center mostly, needs to dry out to become more supportive so that it becomes a harder wood material to help support the weight of the overall tree. Okay, so then this creates those annual growth rings that I was just talking about. And so the secondary growth, it goes through these normal seasons, and we can absorb the annual growth, and we can actually, trace the lineage of this tree and how much growth it's gone through just by counting these lateral growth stems, the lateral growth circles coming from the cambion, where there's the cork cambion, and the cambion there is where we're getting this apical growth. That's where we're developing these new phloem and these new xylem. Now, apical growth and lateral growth, and as we're going to talk with the rest of today, thinking about tropism and how the plant knows which direction it's supposed to be going, it's all going to be regulated by hormones. Now, we haven't really talked about hormones a great deal because the next time hormones really become a big thing is when we do topic 6 and topic 11. But hormones are basically just chemicals that are used by complicated organisms that have multiple types of tissues, multiple systems. They're used internally inside the organism to kind of regulate what's happening, regulate homeostasis, regulate development and growth. And it's kind of a way for different organs and different cell tissues to communicate with each other to cause change. So these little chemical signals are hormones. They get released into the organism, and typically they move around and they only affect one or two target tissues. So they only really cause one or two specific effects. Now, in terms of humans and animals, you're going to learn a lot about different types of hormones. There's going to be a bunch of hormones that you're going to need to know. You need to know where do they come from. You're going to need to know what are their target tissues, what are the things that what's the effect that they cause, right? But for plants, actually, for once, plants are a little bit simpler than animals because they do have a lot of different hormones, but at least for the IB curriculum, there's only really two major hormones that we're ever going to be talking about. We're going to talk about auxins, and we're going to talk about gibberellins. And the gibberellins will come up in the last set of notes when we talk about seeds and flowers and things like that. So for now, we'll mostly be talking about auxins. And so auxins actually is a huge family of hormones. It's not a hormone. It's a bunch of different types of hormones that all fit into this one group, right? And so the different types of auxins that are used cause different effects, right? So they can be used for controlling the direction of root and shoot growth, where we're going to talk about tropism in just a second, right? So the idea of detecting gravity and moving away from the sun, and detecting sunlight and moving towards the sun. They're also going to be there for regulating flowering. So when the plant actually starts to develop flowers, when those flowers are going to open or to bloom, so that they can go through the reproductive part of their life cycle, that's going to be controlled by auxins. The development of fruit after they've successfully been pollinated, that's when things like apples and oranges and bananas and things like that, that's when those fruits will actually develop, right? So that they can help disperse seeds that have been produced from the reproduction. The fruit development is going to be regulated by them. Leaf development as well. The growing of new leaves, but then also the loss of leaves are going to be controlled by things like auxin as well. So in the fall, for example, auxin is going to help regulate when plants decide that it's time to lose their leaves and to drop them, because the days are getting shorter and it's getting colder, and if it's going to snow, they don't want to have all these extra leaves that could catch that snow and weigh them down and break their branches and things like that. And plus, there's no real benefit to having a bunch of energy being put into developing and maintaining those leaves when they're not really getting a bunch of energy through photosynthesis, because it's not particularly warm and sunny through the winter. So that signal that says it's time to start dropping the leaves to the ground, that's going to be helped communicated through auxins as well. And then also responding to wounds, right? Just like you have to, if you get cut, your body has to respond to it, create a scab, try to heal your body. Plants have to do that too. If they get you know, scratched open by an animal, or if, you know, something happens and falls in the woods and breaks off one of their branches or something in the process, they have to respond to that because now they are a damaged organism. And so auxins are also going to play a role as stress hormones to help them react to situations of damage. Now, we're not going to get into a lot of details for most of these, but we are going to talk in pretty good detail about how auxins are responsible for the first one, which are root and shoot growth, and how they help control the plant's ability to know which direction it actually should be growing for its best ability to survive. So the concept that we're referring to is called tropism. And so tropism is the ability for the plant to respond to an external stimuli and understand something about how it should be growing or how it should respond to that environment. So it's kind of like the plant's ability to react. It's like the plant's nervous system in some ways. Like stimulus comes in, it figures out what's the response it should be doing, and then it responds, right? So there are lots of different types of tropisms depending on the stimuli that's causing them. So we're only really going to focus on one, and I'm going to mention another one towards the end. So the main one that we're going to worry about is phototropism. And so phototropism is the idea that the plant is going to respond, and this is positive tropism, by the way. There can be positive and negative tropism. Positive means that it increases it responds in a way so that there's more stimuli or a positive feedback system, right? And then there could be negative tropism where it tries to get away from that stimulus. It tries to move away from what's causing that stimuli. Maybe more, you might think of like a negative feedback system, something like that. So phototropism is this idea of responding to a positive tropism as a response to light, right? So photo referring to light, right? So it's a positive response or growth towards light is what that word is referring to. And you've seen this. You can watch time-lapse videos on YouTube if you want. If you've ever taken care of a plant at home or in your house, you might have noticed that the plants tend to change direction. They tend to grow in specific directions depending on where the light is. And so even though you think plants that don't move very much, if you do watch a time lapse of, you know, plants throughout the day, you can see that they do bend with the light. They do grow towards the light. They change their position in their own environment in order to be exposed to the light as much as possible. That's because they can actually detect where the light is through this process of phototropism. And so the process actually is fairly simple. So what they do is, it's all going to be regulated by auxins, by the way. So around the plants, around the outside, they have a very special layer that can detect the presence of light to a certain extent. It's not the same idea of doing photosynthesis, but it does realize whether or not there is light hitting the surface of this part of the plant. Okay, and this is the coleoptile, sorry. So the coleoptile has this coating around the roots and around the shoots that can help detect the presence of light. And so if light is evenly distributed around the plant, then the auxins, which are going to be produced by the apical bud, right, the very tip of the plant where it's growing, would be evenly distributed. And so auxins are going to influence growth, cell elongation, and mitotic division. So if you have a higher concentration of auxin, you have more growth of the cells, they're going to get longer, and they're going to divide more often. They're going to do more mitosis. So if auxin is evenly distributed around the end of the plant, the part that's growing, you know, this tip of the stem, then it should just grow up straight, or pretty much straight, because all of the compounds are going to affect all of the cells around the stem evenly. So they're all growing at around the same rate. Right? This causes vertical growth. Now, if the light changes, if it shifts from one area to another, one side of this layer, this coleoptile, is going to detect the light at a stronger concentration than the other side. Basically, one side is going to be sunnier and the other side is going to be shadier, right? And so this change in the stimuli causes this layer to shift where the auxin is going. And I'll get into a little bit of details in the next slide, but basically it can actually change through facilitated diffusions through the membrane, right? So auxins have to pass through the membrane to cause this effect on cells, right? It can change where the facilitated diffusion transport proteins are in the membrane. It can make some that are more active, it can make transport proteins that are inhibited, and it can move them to different positions in the membrane, because the membrane is flexible, right? It has that fluid mosaic model. We can move things through the membrane very easily laterally, right? So it will change where the transport proteins are for auxins, so that auxin ends up collecting on the opposite side from where the light is, so the darker side of the plant. And so as auxin accumulates on that one side of the plant, that means that one side of the plant is going to experience more growth and more rapid growth than other parts of the plant. And so that's going to cause the one side of the plant that's not facing the light, right, to get longer and to increase in cellular mass than the other side. So the other side is kind of lagging behind. The far side, the dark side, is going to get longer and longer. And so that's going to cause the plant to bend a little bit. And it's actually going to cause it to bend towards the light. So then now the tip of the plant, right, the apical part that wants to grow as vertical as possible towards the light, is now facing the light better. And so then, because of this, this angle that it's grown into itself, the apical bud is now facing the light more evenly, and so then it goes back to doing relatively consistent growth. The auxin concentrations will go, relatively back to normal around the very ends of the stem, and the stem will keep going straight towards the light. And so because light is constantly moving throughout the day, plants are constantly doing this. They're constantly moving their auxins from one part of the stem to the other part of the stem, one part of their plant to the other, in order to kind of best move themselves or slightly change their positions in order to be facing the light as much as possible, so that they can have the best chance of doing photosynthesis, because really the more exposed to the light they are, the more likely that they can be harnessing that light energy for photosynthesis. All right, so it's really just about moving these auxins to the side that needs to grow faster in order to make the plant bend towards that light source. Now, that's phototropism, right? That's the tropism responding to light. Plants also have gravitropism, which is responding to gravity, right? So plants have the ability for auxins to accumulate because of the force of gravity towards the bottom of the roots. And so as the roots are continuously growing outwards, and auxin, through these transport proteins or through facilitated diffusion, concentration gradients and stuff like that, it tends to be accumulating at the bottom of the root tip, right? So the root tip is where these auxins are being produced, right? Because that's where the meristem is. And because there isn't any force moving them down or up the roots, right? Because gravity is kind of acting on all of these molecules, they end up just accumulating in the roots over time, and so that promotes growth just in the roots. And so the roots continuously grow down, and then they also tend to spread out a little bit in order to find a path of least resistance as they move towards water, in order to spread themselves out through the soil rather than growing up towards the light. So depending on the tissue, whether we're looking at a root tip or we're looking at the stem tips, the meristems in the stem, we're seeing the same hormone being used in order to promote growth in a specific direction. In the stem, it's using detection from light in order to cause auxin to drive growth in one area to make the plant bend towards the light. Where in the root tips, the accumulation of auxin at the very end of the roots is causing them to continuously grow down and out away from the base of the plant, so that the root system gets relatively dense and spreads out through the soil in order to find as much water as possible. Right? So again, it's the same family of hormones, it's just being used in slightly different ways depending on what the goal is of that tissue. And again, regardless of which tissue we're talking about, the whole reason why auxins can regulate this growth is because they're being controlled through concentration gradients, right? So the meristem, either in the stem or in the roots, that is where the auxin is mostly being produced, right? That's the highest concentration of auxin. And so we can guide where the auxin needs to go through controlled transport proteins, right? So if we want to have an uneven growth pattern, like if we're going to bend the plant towards the light source for phototropism, right? The plants can modify how auxin gets into the cells through diffusion, and actually they can even use influx transporter proteins. So they can even use active transport if they need to, if they really need to regulate it fairly quickly, rather than waiting for natural concentration gradients to drive the movement of auxin, they can keep that auxin in one area consistently to keep that growth going through an influx transporter. So using ATP to make sure that auxin stays in that area. Okay, and so there's different types of transport proteins helping out either actively or facilitated diffusion, and so the cell can just regulate where they are and which ones are active and which ones are inactive in order to help guide the auxin towards the area of the plant tissue that needs to be growing the most. Okay, and so they just kind of move in and out of the cell through these influx and exflux transporters. So if we need to move it down, for example, we might kick it out of the cell through an exflux. If we need to keep it inside of the cell, we might draw it in with an influx transporter. But we can use different types of passive and active methods, different types of transporters, and the fluid mosaic model of the membrane, the flax of the membrane is flexible to move these transporters to different directions inside the cell to guide the auxins towards where they need to be in order to help the most beneficial growth. Okay? Now, once those auxins actually get to the tissue that we need them to be there the most, and they start affecting growth, they're going to affect growth through gene expression. So you might remember all the way back, we talked about stuff like this in topic 7, the idea of transcription factors, things affecting gene expression by either increasing or promoting or decreasing or inhibiting transcription rates, right? And so auxin acts like a promoter for genes that are involved in cell growth and mitosis, right? So first off, for auxin to affect that cell, it has to have a receptor in it. So it has to be able to interact with a receptor inside the cytoplasm or inside the cytoplasm. And then that has to trigger a transcription factor to affect the DNA's expression, right? So the auxin binds to this receptor, then that collectively causes an increase in transcription rates in specific genes. So those genes get promoted. And so there's different things that happen, but one of the things that auxin helps trigger is the release of hydrogen ions into the cell wall. So one of the genes that get affected causes secretion of lots and lots of H+. And these hydrogen ions actually weaken the connection of the cellulose fibers. And by making the cell walls weaker, it makes them a little bit more flexible. Because remember, one of the reasons why we're using auxin is to promote growth, right? Specific controlled growth in different directions. So if we want the cells to just increase their length a little bit, right, we need them to stretch. And so we need water to come in, but remember the cell wall is going to be resistant to that. So in addition to helping water move into the cells to allow them to stretch a little bit, we need to weaken the cell walls so that the cell walls are more flexible. So one of the genes that gets affected by auxin allows them to become more flexible through this releasing of hydrogen ions, which disrupts some of the cellulose fibers, right? So in a way, auxins, again, they're going to be affecting gene expression, but there's lots of different genes that they affect. It's not just the cell walls, that's just the example that they want to be focusing on, but essentially they are going to be part of a transcription factor system. And so different genes, depending on the type of auxin that gets released, because remember this is a family of hormones, it's not just one chemical, but it's a bunch of different chemicals. They are going to cause different effects, which ultimately leads to cell elongation and more mitosis. So which is causing this uneven growth in specific locations. And so the last thing we're going to talk about in this, when we think about growth and using growth in science, we actually did mention this before back when we were talking about topic 3, about this idea of micropropagation, or using the plants that can go through the process of micropropagation in order to make like a set of clones. Remember we talked about this in topic 3, this idea of if we wanted to do a test on, how well certain fertilizers work on growth rates, or certain environmental conditions. One of the things that can make it difficult to make our results conclusive is the genetic variation between all the different plants that we might be testing. Even though they're the same species, genetically they could all be different from each other. Well, through micropropagation, we can clone one plant, right? We can make clones of that plant, and so that all of the specimens have the exact same DNA, being clones of each other, we can remove that factor that we couldn't control before for from our experiment. And so the reason why we can do micropropagation is because of this idea of the meristem. If you cut the plant in just the right way so that you can separate new meristems from a budding part of the plant, then when you allow those new meristems to grow in their own soil, in their own condition, then they will be exact clones of the original source that you took them from. And so all of the meristems that you've cut are then going to start acting as cloned plants. And so this process of using one stock plant to create identical clones of experimental plants, this is called a micropropagation, right? And so micropropagation basically means we need to take a tissue sample from a plant and you need to cut it just right so that you get some of the shoot apex, right? So you get some of the meristem from all of these different branches along this main plant. And it doesn't necessarily work with all plants, right? You can't do it to redwood trees or something like that. There's just different types of plants that are stable enough in their meristem content that if you did cut just the apical bud away and the meristem away, even though it doesn't have a root system, it will grow a new root system back, right? So you need to cut the plant in just a way so that it has the cells capable to make new shoots and new roots, even though you cut those away when you collected that sample, right? So not all plants are capable of doing this, but there are various species of plants that can have really stable growth. And so they will be able to handle this type of transplant, this cutting and moving to a new growth medium. So then once we've cut the meristem in just the right way so that it can grow shoots and roots and grow a new plant for us, then we're going to put it on a growth medium. So normally we'll put it on a petri dish or into a special container with a medium, a gel full of growth hormones and nutrients inside, because we need it to go through very, very rapid growth. If it grows slowly, it might not be stable, and it might not survive this transference. So we want to give it the best conditions possible. So we give it lots of nutrients and lots of growth hormones to help along with this rapid growth. Also, when we're going to be doing this process, we're going to be collecting these samples with the aseptic technique. So we need to be very careful when we cut the plant, when we plant the plants into new medium, because we don't want any bacteria to get into our samples, because the bacteria would grow in that nutrient-rich medium, and then they would start taking nutrients from the plants. They might even start to decompose the plants if they're fairly aggressive bacteria, going along doing their decomposing role as saprotrophs. And so the whole experiment would be contaminated and it wouldn't work, right? So we want to also be using aseptic technique to try to make sure that these samples are as pure as possible and they're not contaminated by bacteria. And so eventually, after you get enough growth in your petri dish or your medium, then you can transfer them to soil, and then you can allow them to properly grow to whatever age you need them to be, to whatever size you need them to be in order to do your experiments. Okay? So this is also something that you could consider for an IA, but we don't recommend it because it's difficult. It takes a very long time to get the samples to this point. And even collecting them, I mean, you could collect 50 samples, and of little plantlets, and you might only get like 10 or so that are actually in good shape and haven't been contaminated. All right, so there's a relatively large margin that things don't really work very well for this experimental design as an IA, but it is a technique when you get better equipment, better lab setup, you have better access to some materials. It is something that scientists, botanists are doing fairly regularly in order to do controlled experiments on plants. Okay, so one micropropagation, for example, we could be looking at plant viruses are transported within plants through these vascular tissues, right? So meristems do not contain any vascular tissues, so propagating these plants in sterilized vascular tissue produces virus-free plants. So one advantage to all this is that we don't necessarily have, or we would produce specimens that are free of viral contamination. So then what we could do once we have a clean group that has no viral contamination, we then could expose them to specific plant viruses to see what happens to them, right? The problem with doing that just with a regular plant is we don't know if other viruses have already been exposed to those viruses at the same time. We can't really tell if they already have certain viruses working inside those plant tissues. So by doing this, we create very clean, viral-free plants that we can then test the effects of different plant viruses to try to, figure out what does this virus do to this plant, how does it get into the plant, what tissue layers does it actually affect, and then possibly, now we understand the virus a little bit better, we can figure out a way to treat the plants so that they aren't as widely affected by them. Okay? For example, also looking at orchids as these prized, beautiful flowers. If you have a chance to go to an orchid festival, sometimes Kew Gardens has a nice orchid festival. I recommend doing it. They do look very impressive, very interesting. But they're very difficult to get orchids to breed sexually and maintain certain traits. And so we can use micropropagation to clone the flowers that we want to use, for certain floral arrangements, clone the flowers that we want to keep for pure breeding lines and plants in order to keep the a garden to look a certain way, or for the genetic bank of a certain plant depository. We would use micropropagation to keep those lines going without having them to contaminate each other through mixing different genes through crossbreeding, right? So it's a way to create pure systems for testing, or it's a way just to create pure breeding lines so that they just always exist and that they don't necessarily get modified through natural genetic variation, which is fine in nature, but depending on your career, depending on how these flowers are being used, maybe you want them to look a certain way all the time, and so this is a way this could be done. Okay, so and then this last thing here, looking at basically metabolic pathways, all of these different, this is a metabolic pathway design of hormones. And so it's showing you basically the complexity of a hormone map. And so we're looking at all the different pathways that a particular set of hormones like auxins could follow inside of a plant, depending on the type of tissue that they are being exposed to. So here we've got different hormones. We've got gibberellins, we've got auxins, ethylene is one that's famously used to make fruit and ripen, and there's some other ones. ABA is abscisic acid, which is one I can never say right. Cytokines and cytokinins are ones we kind of mentioned before in other units. And so this look at their pathway themselves, but then also how they're interacting with each other. How sometimes gibberellins can result in JA pathways being activated, which can lead into auxin and ethylene pathways also being stimulated. And so typically hormones, even though they might have one specific effect and have one specific target receptor that they're looking for or that they're going to interact with, they're not necessarily isolated pathways. One hormone can trigger a pathway, can trigger a response in another hormone's pathway. And so that's why studying hormones and what they do and the interactions between different hormones is extremely complicated. Also, it doesn't help that normally hormones in incredibly small amounts, because you don't need a lot of them in order to get a large response in plant tissue or in tissue in general. So tracing how these different hormones are interacting with the tissues can be very difficult because, again, there's not a lot of them moving around, so it's kind of hard to see if it's shifted from one tissue type to another tissue type, causing a different response in that plant. So it's just for your education, seeing how complicated these ideas are. So the thing I've been telling you all this time about auxin, for example, it took a very long time for researchers to be able to understand that's what auxin was actually doing. Because again, it's not just auxin by itself, but there are other hormones that are incorporated into regulating the homeostasis of cells. And so to just figure out just what auxin is actually doing is actually quite difficult. Okay, so that is it for these notes. If you have any questions, let me know.